So, you all see this?
Yep.
Okay, so one of the things that we have mentioned during this uh, coding session series is PCA, or Principal Component Analysis. It's common to mistype or misspell this word, even though it's spelled principal, it's not LE, it's principal. Keep that in mind. Principal component analysis. So if you look up on Wikipedia, the first definition that comes up, it's very complicated, and it causes more questions than clarity. But then you keep reading, and then you find this sentence that says PCA is used in exploratory data analysis and for making predictive models. So ignore that. So far, it doesn't say anything. But what's important is this part. It's commonly used for dimensionality reduction. And by projecting each data points onto only the first few principal components to obtain lower dimensional data while preserving much of the data's variation as possible. So in essence, what this says is when you're plotting things and you have multiple traits, when you have multiple traits, you're limited by how many dimensions you have in your graph. Maximum, you have 3 dimensions, X, Y, and Z. And there might be creative ways to add more dimensions, but that's basically it. And if you have a data set with a lot of traits, then it's very hard to visualize it. And principal component analysis will take a data set with n variables. For example, when we have a phenotypic data set with many phenotypes, fruit weight, fruit shape, prickles, fruit color, leaf length, so on. It's hard to visualize all that at once. So we use principal component analysis for reducing dimensions. In other words, we want to plot the whole thing in 2 axes, X and Y, but then those axes are not actual traits anymore. They're what we call principal components. That means that it's kind of like summarizing a lot of traits in only one axis. And it's trying to show us the variation between different individuals or different accessions. We can do PCA in R. I can show you how it's done. But before we start on that, I have to make sure that you all have access to the cluster. If it doesn't work, we can just do it in RStudio, but it's better to do this kind of analysis on the cluster because they take a lot of space. So pause this recording real quick. Great. So I added you all to the cluster. You can do this. You can go on your computer and find the terminal. I'll show you in a second. In the bottom, I usually have it open, but you guys might have to go to Launchpad or search up here from this option. Just type terminal. And the first one that pops up, you open that, and I have multiple connections here, but yours probably gonna be just one. And then you click on this plus button and helps you connect. And what you need to type is SSH and your ID or actually that doesn't, it's not even necessary. You can just type this and it says, do you trust the server? You just type yes, and then you put your password in it. But it's better to type your user ID here. Uh, so user ID at, instead of user ID, you type your actual user ID and then at this address. So I'm gonna copy this into the chat.
Hamid, what
Yeah.
does SSH mean again?
SSH is a form of connection where you're remotely connecting to a server where you can do some tasks. So in my case, it will be SSH HR355, and we hit enter, then it's gonna ask for my password. This password is the one that you guys used for connecting to the cluster.
Mm, it didn't ask for my password.
Ah,
Yeah.
can I see your screen real quick? Wait,
Oh,
sorry.
you said
Wait,
it.
wait.
Yep.
Yeah. Okay, go ahead.
Yeah, could you see it now?
It's did you hit enter?
Yeah, I did.
It's not connecting, looks like. Can you close out of this? Oh, close out of the program and start over.
Okay. Yeah, it doesn't work. Is there supposed to be another command before that? Oh,
Can I see your screen again?
okay.
So SSH, this looks correct. Does anyone else have any issues?
It won't let me
Oh.
type in my password.
Oh, okay. I think I know what the problem is. You guys are off campus, right? Is everybody off campus?
I'm off campus.
Who else is?
I'm in the
Anybody?
library.
Okay. So if you're on campus, it should work. Do you work, Eddie, for you? Will you stop? So for off-campus people, sorry, I have to share something real quick. For off-campus people, they need something called Cisco AnyConnect. So you have to set that up first.
Okay.
And so you type Cisco AnyConnect for now, and then it shows you the VPN. You download it for your machine and then install. And then you have to use the duo push notification on your phone to get access to this. This is a very useful tool, by the way. It allows you to, if you're off campus, it allows you to use Cornell services, such as the library, as well as the Google Scholar subscriptions that Cornell has. So if you need a paper, chances are that off campus, you won't be able to download it. But using this VPN, it will connect you to the Cornell servers where you can download stuff.
Ahmed, can you go through
Yeah.
the login process for BioHPC one more time? I had some trouble
Sure.
getting my terminal up.
If you share your screen, I might help you better.
Okay.
So you type SSH, your user ID, no, just user ID at no, at the address that I put in the chat. Okay, get rid of one of those ads. Try that. Yes. Awesome. Your password. When you type it, looks like nothing happens, but it's recording your password. There you go.
Okay.
Your
Nice.
it's logging in. If you type ls now, see what happens. Just ls. Okay, cool. And then we're gonna work in the work directory. The address for that is cd space / work dir. Just lowercase. Okay, that's where we're gonna be at. And you can wait there for us to get there, to meet you there.
I think there was one thing I had to do. It
Mhm.
wanted me to make a password, and it sent me like
Right.
in the email. It had me do. There was like a little 15 slide show I had to look at, and it.
Oh, right. Because it's your first time.
So I don't know if
So
maybe that's for other people. It's not letting them put in their password because of that.
Yeah,
point.
I've done that already. And then, oh, but I can't even connect. I'm trying now, but it's still not letting me.
Right.
I already set up the dual mobile for the.
Okay.
For the
Paul, can you show me what's going on your computer?
oh, okay. Like, I think yeah. So if
Okay.
I go here,
Did you download this stuff? Okay, go ahead.
yeah, they want me to.
It should be something you put on your computer, right? Did you put on the Mac thing? And for Duo Push, you have to go on your phone. Do you have the app?
Yeah, I do.
Duo Mobile. So let me show you on the app.
Mhm.
If you open Duo, let's see. They make it super secure, and this is a new thing. In the Duo, it should, under Cornell University, it should give you a password.
Oh,
And
yeah.
that's the key you use. If that doesn't work, then you refresh it.
Okay.
So
Yeah, it's still not letting me.
let's start over. Try download. So Google, you can keep your screen on so I can see. I think the other people should also download, even if you're on campus, you should download Cisco because chances are that you might be traveling or anyways, but this is good information for everybody. Paul,
Okay.
so just type Cisco and you connect Cornell. There, first one. Okay, and wait a second, okay. So installs here, okay. We have to install it first somehow. I just don't know where the software is. It should give you the actual software that you need to install.
Yeah, they bring me when I click this link to go and install, they bring me to this page.
Let me go back to that page. Let me Okay, a downloads page will appear. Okay, try again with the CU VPN, yeah.
Okay. Yeah.
And this password is your Cornell password. It's not the cluster one, by the way.
Yeah, it's not letting me log in.
Oh, did you refresh the push passcode thing?
Yep.
Which push are you using?
The Cornell one, but
Uh-huh.
I've tried both and it doesn't work.
It might have logged you out, unfortunately. Oh, there you go.
It's so weird.
Yeah.
Maybe they just
Okay.
want you to try many times.
So is life. So now, yeah, you have to be able to easily install.
Sorry, so the password is the same with our is the Cornell,
Cornell,
and the
right.
dual passcode is a software passcode.
Duo, Paul, can you explain how to use?
It's when you use your Duo mobile. Yeah, it's like this. Is this one over here? Then this is that third party thing is the cluster one. So you use your normal Cornell net ID and password, and you key in that one to get to the download page.
Oh, okay, thank you.
Were you able to get the software going?
Yep. I'm installing it now. Should I stop sharing first?
Sure. Does anyone else have any trouble? All good? If you're, Eddie, were you able to get Cisco?
I'm downloading that right now. And I was able to log in the terminal, so I'm set.
Cool. Yeah.
Can we use this VPN in other country?
Yes. That's the
Okay,
cool
cool.
thing about it. I remember when I got accepted to UConn. So back home in Iran, we don't have access to a lot of papers, unfortunately. So, and our institutions don't have a lot of money, so they couldn't buy the subscriptions for the journals. So a lot of these papers that we commonly read and we don't even think about at Cornell or other US institutions are not available in a lot of countries. So when I got accepted to UConn, they gave me the user ID and password, and it just changed my life. Even before I got to the US, I was able to connect and download papers. And I used to download papers for people in my entire dorm, like chemistry people, engineering, who want to come get a paper from me. I was a paper dealer. I should have charged them. Runlan, is it working for you?
I just downloaded.
You downloaded Cisco?
Yep.
Okay, do you need help?
No, actually I am on campus now, so I'm not really.
Okay. Were you able to connect to the server?
Yeah. So you mean like from
Okay.
the terminal, right?
Right.
Yeah, I can.
Okay, so we can continue this on the server. So you guys all have terminal, and the other software that you guys are gonna need, probably not for today, but something that you're gonna need is called FileZilla. And in that 10-page instruction that they gave you when you signed up, they should have given you some information about FileZilla. That's a software that is used for transferring files between your computer and the cluster, whether you want to upload or download. And but I'll quickly show you what it is. So FileZilla looks like this. FileZilla. Wait, what's going on in files? Sorry, it's getting on the full screen mode and I'm not able to get out of it. Okay. Anyways, so one side of this page is my computer, which is here, all of my files with the location under my stuff analyses and blah blah blah. And so I label my analyses these ways, like I said, with the date and the kind of analysis that I do. And all of the folders on the cluster are here. I'll show you how to connect to the cluster on FileZilla. So this is going to be recorded. You can do this later. But I'll quickly show you when you click up here in this icon, the site manager pops up. And there you can create a new connection. A lot of people use this option, host username password, quick connect. I don't recommend that because that means every time you want to connect, you have to redo it. But here you can actually save it so that you can always use it in the future. So I made a folder for Cornell. I connect to Cornell servers a lot, so I want to save all of these. And then I clicked on new site. And then there I typed the host address, which is the one that I gave you in the chat. The CBSU workstation 3. That's CBSU workstation 3 at .biohpc.cornell.edu, and the protocol is SFTP, SSH file transfer protocol. That's all you need to do, and then your username and password. Okay, and then you can, I believe you have to give it a name. Let's see what's going to happen. Okay, now it's connected. But then this one, I want to change its name. I want to call this CBSU WRKS T3. And then it will always remember that. And so here, what we have, it says remote site. You can type the address, work there, work directory, and that's where we are. We're gonna do all the analyses that we do today here. And then if I wanna transfer any files from my computer, I can just go into the file and say, right click on it. If I double click, also it will upload it. So I just double clicked on this file and it went into the cluster. So to double check that worked, I can go to the work directory. And this is the file that I just uploaded. So that's a quick intro to FileZilla. That's something that you can install later, but today we're gonna mostly use the terminal. We're gonna try to do PCA in the terminal. But for PCA, I have to give you guys some files. So, okay. Let's go to stop sharing for a second. find those files. Okay. All right, let's see what we have here. We need a GDS file copy. Into, let's see, what's the best way to do this? Into my home, I guess. So if you remember, this is the sign. This sign means home directory. And that's where you can see your home directory. So we have two directories here. One is called work directory, where we run analyses. But then when we finish, we save all the results in the home directory. So I'm on a different machine right now. So I want to copy this file that we need for PCA into my home directory, where then I can copy it into this machine. So if I type ls home star GDS, that's my file, and then I can just copy that into here. So I was just able to transfer one file from one server into another server by copying the file into my home directory first, because the home directory is shared in all of these servers. While that's copying, you guys can go ahead and make a folder for yourself. So I'll start over. SSH. Here's the chat. SSH. Chat. Oh, like that. Okay, and then cd/workdir ls. We're all set. Here now we have to make folders for ourselves. You can just give it your first name. I call it Hamid. You can call it anything, but to make it easier to distinguish, just give it your name. So you make a folder for yourself. Are you all there? Any questions so far?
Are we all on the same? We're all on the same server, right?
Right. So the server name is this one, CDSU Workstation 3, but we're going to run the analyses separately.
Okay. I was just wondering because I saw Hamid and Eddie now as folders, and I was like,
Right.
whoa, how did they do that?
So yeah, we're all collaborating now. So I see 3 people, Paul and Roman.
when I do cd/workdir, it says no such file or directory.
Sorry, something is going weird in my computer. Let's see what's going on here. It's not letting me see you guys. Weird. Okay, so show me your screen. Okay, you have to go to workdir, cd/workdir. You didn't type it correctly, Chris. No,
Wait.
no. Wait. cd workdir, work.
Oh, WORK.
Yeah,
Oh,
okay.
oh, whoops.
No
Oh,
problem.
nice.
Now you can do make the mkd dir. Okay, that's all you need to do. Cool,
Okay, thanks.
sure. Roman, are you all set?
Yeah, can you see my directory now?
Okay. Yeah, okay. I see everybody's directory. Let me share again. So now, okay. Anyways, so if I type ls, I see everybody's folders, or I should say directories. And then what you need to do is copy this file, all variant x math2p.gds into your directory. So it's going to be the same up to here, and then instead of this, you type your directory. So for me, it's /Ahmed, which is going to be /Diego, /Eddie, and so on. So I just copied it into my directory, and then I go inside my directory and type ls, and I should have a file that says all variants except math2p.
It says permission denied.
Permission denied. To copy?
Yeah, wait.
Oh, okay. I have to change the permission in this work directory. Let's see. But it's weird. It allows you to make directories, but okay. Let me stop sharing. Oh, can you share?
Yeah, might have entered the wrong code.
Yeah, it says the same thing for me.
Paul, are you trying to share?
Yeah, I'm sharing. Can you see?
No. Weird stuff is going on in my computer. There are so many things open. Let's see. Okay, now I see it. Okay. So yeah, that looks good. Cannot open for read. Okay, I should give permission to everybody to use that.
Okay, I'll stop sharing.
Yeah, thank you. So I have to quickly look up. I can't remember exactly how to do this, but I'll look it up. What I need to look up is Unix change access options, read, write files, manage file permissions. So I think this is what I need to do. Okay, try now. Try copy.
Same thing.
Same thing.
Permission denied. Yeah.
Okay, let's try this one. You should try again.
Still the same.
Sorry, this is weird.
Yeah, noise.
Yes. Right. Try now.
Permission denied. Same thing.
Weird, weird.
Hmm.
I,
Is it something like me being off campus still?
no, I think has nothing to do with that. Do other people have the same problem? I'm pretty sure they do.
Yeah, I have the same issue.
Mm-hmm.
Okay. Let me pause this recording real quick and find a solution. For people who are gonna watch this later, what I did was chmod a+r and then the file name, and this gives permission to everybody to copy that file and use it. So now we can go to our specific directories and make sure that the file is there. Okay. And if you type R now, just R, capital R, it should take you to R. So R is already installed on the cluster, and it already has a lot of packages in it. So many other things that I need to use, it's already installed, but sometimes we have to install some of the packages ourselves. So if you type R, you should see something like this. It tells you the R version, 4.05. When you write a paper, they're going to ask you what R version you use. So in the methods sections, you have to add this. And then they give it funny names usually every time they have a new version. Every 6 months, I think they have a new version. So one package, before I continue, does anyone have any questions, trouble? Are you all at this page where I am?
Yep.
Cool. Runlan, all good?
Yep.
Awesome. So I wrote a script a long time ago, several years ago actually, for doing PCA and phylogenetic analyses in R. It requires two packages, GDS FMT and snip relate. I'll copy the commands into the chat and into here. So if they are already present on the cluster, they should load. Now it's still loading. Okay. So before I start doing PCA analysis, what I did was I converted our SNP files, the variant files, into what's called the GDS format. GDS format is a specific format for this package, SNP relate, and GDS format. But I'll show you what the file looked like when we were in the VCF format. I'll quickly show you that before we run this analysis. Let's see. So I did variant calling. In variant calling, I did it for each. Basically, these are the files that shows how each accession differs from the reference in what positions. And then when you combine it all together, this is the file. And then I did another filtering, removing the rare alleles. So if there is a SNP that is only less than 2.5% of the individuals, then I didn't keep that SNP. So I got rid of it. That's what math 2% means. It means minimum allele frequency below 2%. So if a SNP has minimum allele frequency below 2%, then I didn't keep it. It was just a way to simplify this analysis. So originally I had about 20 million SNPs in this one, and then we downsized to about 10 million. So,
And just a quick question,
yeah,
Ahmed.
sure.
By removing the SNPs that are at a lower frequency, those could be just holes in the quality of the sequences and other things like that, so it's easier to remove it.
Right. So a lot of people remove those because of the quality reason, like you mentioned. Sometimes, if it's rare, chances are that that's actually an error. But also for computational reasons, if you have too many SNPs, then some analyses might not work. So you downsize a little bit so that you can do your analysis.
Okay.
So if I type zcat, the file name now, and then more, type more is very important because if you don't do that, it will open the whole file. And this is showing the top part of what the VCA file looks like. A lot of the pound sign, hashtag, these are all comments. Just shows you what chromosomes we have, length of each chromosome. Chromosome 1 is 108 million base pairs, and so on. This one is 77 million, up to chromosome 12, and then information on what indels look like and the accessions we have. So this is doesn't have line breaks, so it looks a little weird, but you could do, let's see, the larger screen, it looks better. So first line starts here and goes all the way here, but this display puts it kind of in separate lines. Anyways, so you have chromosome, it tells you which chromosome, chromosome 1, position, 338 ID. Doesn't sometimes in the human genome they give IDs to SNPs because they want to know if, for example, one SNP causes a disease, they want to have a unique ID for it. And then it says reference is C, An alternate allele is a T, and then a quality score. And remember, we were talking about 0/0 means that this is homozygous for the reference, meaning that this accession, the first accession, MM00097, it's just like the reference and it's homozygous. In some cases, you might see 0/1, which means heterozygous, or 1/1, for example, in this case, that means that it's homozygous for the alternate allele. This one. And this just keeps going on. Right now on this screen, you're seeing 1, 2, 3, 4, 5 SNPs. And you can just scroll down and see, it will also show you the indels. And basically you can just scroll down forever because there are 10 million SNPs. But we don't want to do that. I just type Control-C to get out of that. I just wanted to show you that. There's a software called Plink that can convert VCF into another format. And then from that format, we convert into GDS format. And that's what we use for, so this file that I gave you is the one that we need to use for PCA. I understand it's 1 p.m. If you guys need to go, it's okay, we can stop and continue next time. But if you want to stay, I'm happy to go on for another half an hour or so.
I can say.
All good?
Yes.
Diego and Bruno? Okay. So, yeah, let's do PCA real quick. In your R, so we were here. What you just did was load these two packages, and you type geno file, this, which could also be, if you remember, I said this one, sign, which is kind of like an arrow, can be replaced by equals. And that's what you type. I'll put it in the chat also. And if nothing happens, that means that you were able to open that file successfully.
I made a
Stand
quick
up.
question on the SMP calling.
Yeah.
On the ones that had, so you showed us ones that had 0, 0, 0, 1, or 1, 1. There were some that just had dots. Is that just missing data, or?
Yeah, that's a good question. Dot/dot means we didn't have any reads for that, or maybe the reads were low quality and we removed that site because we weren't sure about it.
And so the data still lets us know that there was an SMP there, either that the data wasn't missing or was lower than 2%, but it still wants to let us know that it found one there.
So if it was lower than 2.5%, it wouldn't show up. If it shows up, it means that we just don't have data for that individual specific, because we have data for the one next to it. That means, for example, one of those accessions had data, but like many of them have data, but some don't.
Okay.
Only in that specific individual we don't have.
Mm-hmm.
Good question. Any other question? Okay. So let's get back to here. Were you able to open the geno file, which is the GDS file that you guys copied from working directory? They work? Okay. So the next line of code is just to get the SNP IDs and sample IDs. I put in the chat. So SNP ID equals read GDSN index it and find the SNP IDs and sample IDs. So sample IDs are just accession IDs. You can type head parentheses SNP ID, just numbers, and the sample ID looks like this. And if you type sample ID, it will show you all of the accessions you have in this file. Okay. Now we get to actually run PCA. One difference is that here I used 64 CPUs to make it fast, but the machine that I reserved for you all has only like 16 or so CPUs. So this last number of thread, you're gonna change it to 1. So this is how you're gonna run. In the chat, I put the command. I'll copy paste it here and I'll explain what it does. SNP GDS PCA is a function that takes your genome file and it needs the SNP ID, which we just made, and autosome only, false. Here, in the human genomics, we have the sexual chromosomes and autosomes. This option is written for those people. If they don't want to include the sexual chromosomes, they can use this option. For us, we want to use all of the chromosomes. So we just say autosome only false. Remove mono SNP. If there's a SNP that is only in one individual, we'll remove it. This option for us doesn't matter because we have already removed the ones that are below 2.5% of individuals. So we don't have any SNP that is present in only one individual. And finally, number of thread equals 1. And if we hit enter, it's gonna start running PCA. It tells you that we have 70 samples, 11,972,680 SNPs. And using one thread, meaning one CPU core, and now it's running principal component analysis. And here it's showing you how far through it is. So it's still 0%, but in some systems it actually shows you in real time the progress, but here it just showed you it went from 0% to 100%. So it finished. But for some reason, it wasn't showing us the progress. Anyways, so it says that it made the eigenvalues and eigenvectors. These are some jargon related to principal component analysis. And then you can look at the eigenvectors. I'll explain in a second what that means. So these are the vectors or the axes that principal component analysis converted our data into. So instead of having like SNP data, we get these numbers, just the differences. And then one thing that is very important is the percent variation. So if you type this, PC.percent PCA variation proportion times 100. So these two numbers are very important, and if you write a paper and do PCA in it, you have to report these numbers. That means that the top components, top principal components explain 23% and 12% and 5% of the variation that is present in your data. So one axis, the first axis, can represent 23% of variation in your data, and second one 12. Obviously, if you add all these up, there will be 100%. And quick note about why we put things in parentheses. If I type this without parentheses, it will just show nothing. It will create a new object and save the results into it. But if I type it inside parentheses, it will show me what it does. So the output from the command is displayed. That's the difference between putting the command inside parentheses or not. That's not very important. I just wanted to explain why I did it like that. Now we want to simplify. We want to make a table called tab. And we want to create a data frame with the accession names and the eigenvector 1, eigenvector 2, 3, 4, and 5. And those eigenvectors or principal components are the ones that I talked about. Those are the axes that explain the variation in our data. So if we type that, then it's going to create a table for us. I'll give you the code. When you copy and paste, you should be careful. There might be issues with copy paste. If you run into trouble, let me know. You can actually type this code in one line to avoid errors. So if you run into errors, just make it into one line. But you have to make sure that you don't delete anything. I'm gonna give it to you in one line. Did it work? If you type just tab and press enter, you have to see you have to be able to see something like this with the accession names and EV1, which is eigenvector 1, eigenvector 2, 3, and so on.
Hey, Hamid, can I share my screen real quick?
Sure.
So I think I was having some trouble with the files not being present
Okay.
on the computer here. So when I went, so after I checked out the sample ID and showed all the extensions, I tried to run this PCA that you put in the chat, and it gave me this error. It said that the SNPID.id is not found.
Okay, if you just type snip.id, okay, that looks like that command didn't work for some reason. You can just copy from chat the line that says snip.id read.gdsm.
Okay.
It's before sample ID. You can do both sample ID and snip ID.
Okay, I'll do the snip one first.
Okay. Okay, cool. If you, I think accession sample ID is okay. You can just type it. Type sample ID. Dot ID. Sorry. Okay, that one is good. Now you can run the PCA command and it should work. Okay, that'll take about a minute. Does anyone have any questions? It's very important to point out that I want to make sure we're not just copying and pasting code. We have to be able to know what each part of the code does and whether we can modify it if we run into issues.
I'm still a bit confused as to like what's the advantage of running PCA on SNPs? Because I was under the impression that PCA was more for like phenotype expressions.
Yeah, you can do it both on phenotypes and SNPs. For SNPs, it gives you a quick visualization of how diverse the groups are and how far apart they are. And by the end of this exercise today, we can make a plot and then look at what the wild ones look like in terms of variation, what the cultivated ones look like, and we can color code them to separate. Some people also use machine learning with PCA to identify, let the program choose where the major groups are, where the wild one ends and the semi-domesticated one starts, or where the semi-domesticated one ends and cultivated one starts. So those are some of the uses.
Okay, thank you.
Sure.
Was the terminal supposed to show anything once we ran that line of code? It starts with tab.
If you then just type tab, it should show you the contents of the table that we just created. So if you just type tab like this, it should show you this table, which has the accession names and 5 columns. It's printing one of the columns down in the bottom here. Are you able to see that table?
It works. Thank you.
Oh, cool. Sure. Okay, we have done the PCA now, and we have made a table. Next, we need to load our populations. So in this population file, we have the accessions as well as their identification. Like it tells us the accession as well as whether they're melongena, ovigerum, or insan. But I haven't given you that file. I should give it to you real quick. Let's see. Okay. So there's gonna be a CSV file that you can use. You on your terminal, you can make a new connection so that you don't lose the work you have just done. So on your terminal, there's the option to add a new connection to the same server. So it's just 2 separate screens, but it's the same server. And we're gonna have to copy a file called populations. Let's copy CSV to here. Search mode. Let's see if I remember what the CH mode was. And I'll copy it for everybody, actually. You don't have to do anything. It doesn't let me. um, okay. It probably doesn't let me because I don't have access to your directory, which is weird. You guys can try doing this on your own. See if you can get the CSV file into your directory.
How do we get out of the R prompt?
No. Keep that on. We need that. What
So
you need to
open-
do is click on this plus sign and make a new connection with SSH. Pause.
Yeah.
Okay. No, it's not that. Let's see. It should show you somewhere, or you can just start a new terminal actually, see if that helps.
Okay.
Or in the terminal option at the top, it should let you open a new one. Let's see if that does anything.
Mm,
Oh, I just went to
Oh.
shell.
Oh, wait, there.
The new tab.
Yeah.
Okay, sounds good.
Sure.
Mm,
And so now in our directories that we made within this server, we should have the variance file and the population CSV
Right.
file.
Right. We're almost there, guys.
mhm.
We'll do this today. Determine we do PCA. We did PCA, we just have to plot it now. But the advantage of what we did today is that we learned how to make a shared space on the cluster. I'd never done that before, so it's cool. We can now work together and run GWAS in the future on the same space. Are you all set? Everybody?
Yep.
Paul?
Oh wait, I'm still trying to connect to
Okay.
something.
All right. Are we recording? Let me double check. Okay. It's like we are recording. Nice. So back to PCA, we need to open that CSV file that I just gave you guys. To open it, you type populations equals read.csv and then the name of the file. Chat. Except I didn't do it. Everybody did this but me. Fresh. The easiest way to transfer files is actually to do it with FileZilla. With FileZilla, you can literally just drag and drop stuff. There. Okay. So try again. Now I have a populations file. If I type its name, it shows me what's inside it. It has 3 columns, the accession names, the population. Right now, we don't know the specific populations, but we have a general idea about their IDs. And if you remember from previously, I said that some of these names are questionable. So people call them Ovigerum or Melongena, but they might be actually wild, for example. So that's something that we can check with PCA and also phylogeny. With the same package that we're doing PCA, we can also do phylogeny. The tree that I showed you guys last week, I made it here, but I'll tell you how to do phylogeny next week. For today, we can just finish the plotting. Were you able to open the populations file?
Hamid, I cannot open.
What does it
Can
say?
you-
Can you? Sure.
Can I share my screen?
Yes.
Yeah, see it? It's on my left.
Sorry, my screen is going crazy here. I don't know what's going on with it. Just one second. Wait.
Yeah,
You-
see.
Okay, in the copy part was not inside ours. That you should copy. So go to your other tab. Okay. The type cd/work directory /work dir. dir. Oh, ls. Now CP. Let me wait a second. Wait. Let me do something on my end. Okay. Now ls again. Okay. Now CP populations.csv space room.
Is that right?
Yeah. Oh, again, the same problem. Try now. Just the same code. You can press the upper arrow. It will bring back the last code you typed. There we go. Okay. Now you go back to R. And in R, you type the last code that I copied into chat.
It's in the chat, right?
Right.
Okay. Thank you.
Sure. Let me know if it works or it doesn't.
It works.
Awesome. So back to PCA. This is our populations file. And we're gonna merge the table that we created previously. So the table we created has just the PCA information, and we want to give it the accession. So we learned previously how to merge 2 things by 1 column. In this case, the column that we use for merging is called accession. So we're merging tab and populations by accession. And I'll give you the code. Okay. When you do that and type tab, then it adds the IDs for all of the accessions. Okay. And this part of the code says delete the population column. We just don't need that right now. And this part of the code says save the output into a file, write that table, tab, and call it PCA output txt file. So it will create this file based on this tab. We're saying that we want to keep the column names, but we don't want the row names. Row names are just numbers. And quote is false. We don't want quotes, and we want the separation to be tab. If you remember, I said that separation can be comma separated like in CSVs, or tabs or spaces. Tab is the most common way to separate these columns. That's it with that. And then we just want to give it a color and symbol. Right now, we want to create a column that says color and just put zeros in there. This is just the empty column for now, but we want to give it different colors. And the color is going to match the IDs. For example, Melongena, Ovigerum, and Insanum, they're all going to have different colors. And I'll give you these codes, these lines, symbol. The next one is population names. We want to define a vector. We want to say what populations we have or IDs we have, Lasicarpum, Insanum, Ovigerum, and Melongena. And what colors. You can use any color, and there's a guide on Google. You can search R color guide, and it will give you all kinds of names, but these are the ones that I've used, and they worked. So you can use that, pop colors, gray, green, yellow, purple. And then this line of code says, delete the ones that are NA. Is NA means if something is equal NA, like if any of the IDs are unknown, and exclamation point reverses that. So basically here it's saying keep all the ones that are not NA for the ID. So tab equals tab, where tab ID is not equal NA. That's that. And then we're doing a for loop here. So for each population, we're saying if the ID equals a specific population name, we give it a specific color. Like the first one, Lasicarpum will be gray, Insanum will be green, Ovigerum will be yellow, and Melongena will be purple. This is a for loop because it's repeating the same accession with different values. So we're saying for I in 1 to length of pop names. So pop names, their length is 4. So if you type length pop names, it should be 4 because we have 4 IDs here. And it says for I in length of pop names 1 to 4, find inside the tab where the ID equals pop names 1. Pop names 1 is Lasicarpum. So it's finding all the places where the ID equals Lasicarpum, and it changes the color into the pop colors I, which in this case will be the first one, pop colors 1. I understand I haven't explained for loops, but I'll do that in the future. But for now, just to understand how it works is enough. So if I type tab now, it should have a symbol column and a color column. And each accession has a color depending on its ID. All the Melongenas are purple, all the Insanums are green, and all the Ovigerums are yellow. And we have some other ones that we have also given a color to, like Lasicarpum, which is great. Are you all there? Okay. Now the last part is we can make a PDF. So that's quite easy. In this part of the code, we just give it a name .pdf, and it can be anything. And we define its width to be 4.5 inches and height also 4.5 inches. And you can change these numbers. These are arbitrary. If you want a bigger plot, you can make it bigger by changing these numbers. And then we want to plot eigenvector 2 in the table and eigenvector 1 in the table. The x-axis label will be PC2. The color will be the same as the colors that we just defined in the table. The y-axis will be PC1. PCH is the symbol. For now, we're using empty circles. And these are some information about the names. I actually can't remember what this frame plot means, whether you want to have a frame around the plot, which I say I don't want. And LAS is the orientation of the text. So you can give it a legend too, but here I decided not to do that. And if you copy paste this, it should give you a plot. So you guys won't be able to see the plot because you don't have FileZilla, but I'll show you what it looks like, and I can actually go into your folder from my computer and tell you whether it worked or not. Did you all do this part?
Yep.
And the last part is dev off, meaning we want to turn off the device. That means that the visualization device where we put the plots, we're telling the program that we're done using it, so we want to stop making plots. That's what it means. So I'll go into your folders real quick and see if it worked. It's not letting me, of course, but you can go into the other tab that you created and do this. Let's go to your directory, and if you type ls, you should see this PDF file. And for the naming, I named it PCA 2-1, meaning the second eigenvector and the first one. And this part is just the date. Okay. Do you all see that PDF?
Yes, I do.
Okay. So if you have FileZilla, that PDF will show up like this. And then you can download it somewhere on your computer and then open it once it's downloaded. So this is the PCA that we just created. We call this a funnel shape, where we see a large range for the wild ones. All the green ones are the wild ones. And then all of the cultivated plants are clustering together. What that means is that the genetic diversity is low among the cultivated, and it's very high among the wild. So we're seeing a bottleneck effect where genetic diversity is kind of on this plot. It's kind of like a triangle or kind of like a funnel where it gets narrower as you go from wild to domesticated. This is called a bottleneck effect, and it has been seen in many crops. And also other domesticated organisms like horses, dogs, etc. And you go from wild, which is very diverse, into the semi-domesticated, where all these yellows are. And then those have medium diversity, and then a cultivated plant that has very little diversity. And then you see some like purples that are between the yellows, some purples that are very close to the wild, and then you start questioning whether those are actually what we think they are. They're called melogena, but we can check their fruit. I've been checking some of these fruits and seeing that they're not actually domesticated, more likely ovigerums or wild ones. And some of these wild ones are coming in the intermediate group, so likely that these are ovigerums. So this can be useful for that. Also, the phylogeny that we can talk about next week.
I have a question. So,
Yes.
why do we do the analysis on the terminal instead of using the R?
Yes, good question. The GDS file that we use today has 10 million SNPs, and it's and before you make that, you have to actually use another file format, which is the Planck format, that is the intermediate step from VCF into GDS. In other words, those files are huge, and if you open it on your laptop, chances are that it's going to crash because it requires a lot of RAM. Actually, you might get an error saying that this file is too big and it won't be able to work. And so that's why I decided to run it on the cluster. The other advantage of running things on the cluster is that you can access it anywhere. Like the files are there if you're off campus or anywhere, and you can download it from there. There is a point in your bioinformatics career when you realize that your laptop is useless, because no matter how good it is, even if it's like very advanced, still nothing compared to what the servers have in terms of CPU and RAM. So that's why I encourage everyone to use the cluster.
So basically, if we're using terminal to do the analysis, we're not doing the analysis locally, like on your computer.
Right,
We just
exactly.
send instruction from our computer to something. Maybe I
Right.
don't know what's the terminology for it. Something like cloud, like
Yeah, this is the servers are actually a form of cloud, and we're connecting to cloud and running analysis.
so physically there is a supercomputer on campus doing the job.
Right. Yep. I don't know where it is, but I know it's somewhere on campus. We can figure it out if you're interested. I was told once that people go visit these supercomputers and they get really excited because they see these huge floors with tons of computers connected to each other.
Interesting.
Kind of like FBI servers. Eddie and Diego, you guys are going to stay online. Are you still available? Good. Cool. Anybody, any questions before we end the bioinformatics session?
I'm good, thank you.
Okay, great. It was nice seeing you all.
Yep.
We're gonna switch gears and start talking about Eddie and Diego's presentations. I'll stop recording.